Because of the migration of these cells, three derms are formed. That is termed as ectoderm, mesoderm and endoderm. The cell from the epiblast when migrate or form primitive node, then some cells of the epiblast detach from the primitive state and then it moves downward and mingle with the hypoblast. When the cell mingle with the hypoblast, these hypoblasts do not take part in the formation of any germ, but it is considered as the playing important role in the formation of primitive state as well as in the formation of portion of yolk cell and the formation of yolk stalk that connect the mass of the yolk with the embryo. Here is the formation of derm. How endoderm is formed? Because of the migration of some epiblast and mingle with the hypoblast, in the result of which endoderm is formed. While remaining cells of the epiblast migrate to the primitive state into the lateral side of the blastocoel, in the result of which mesoderm is formed, while the remaining part of the epiblast form ectoderm. Here are different views of gastrulation, in which you can see that some cells from the epiblast move downward and attach with the hypoblast, in the result of which endoderm is formed. Hypoblast form here some portion of yolk cell, while the remaining epiblast to the primitive state move laterally into the blastocoel, in the result of which mesoderm is formed. This is the frontal view of gastrulation, while it is a lateral view of gastrulation, in which you can see that on the posterior side to the primitive state, cell of the epiblast move laterally, that is the posterior part of the primitive state, in the result of which mesoderm is formed, while with the hypoblast, epiblast mingles, in the result of which endoderm is formed. This is the endoderm. While anteriorly, these cells move and form nodochord, that is present inside the Hansen node. Hansen node is the soil portion of the primitive group, and in this epiblast move forward and form nodochord. It is the upper view of gastrulation. Here is the primitive state and here is the Hansen node. When the epiblast move inside the Hansen node, in the result of which notochord is formed, while when it is moved laterally, then mesoderm is formed, while with the hypoblast endoderm is formed, while the remaining cells that do not migrate through the primitive state, it forms epiblast. Here is the complete view view of the formation of ectoderm, mesoderm and endoderm. Here is a yolk sac that attached with the endoderm, and this yolk sac is attached with the embryo through the stalk, which is termed as yolk stalk, that supply nutrients to the embryo during development. The cell that migrated to the lateral side of the blastocoel is formed mesoderm. Mesoderm later on divided into three regions. One is the paraxial region, which is also termed as epimere. This is the epimere or paraxial mesoderm. Then here is the intermediate mesoderm, which is also termed as mesomere. While the third region of the mesoderm is divided into two parts. One is the somatic mesoderm and other one is the splenic mesoderm. Here is the splenic mesoderm that surrounds the embryonic sac, while here is the somatic mesoderm that attached with the ectoderm. When the splenic mesoderm, or which is also termed as splenic pleury, when attached with the endoderm, in the result of which visceral layer is formed that later on form different visceral organs. While somatic mesoderm or somatoplury, somatic mesoderm is also termed as somatoplury. This somatoplury, when attached with the ectoderm, in the result parietal layer is formed that later on form the parietal body wall. In the splitting of these hypomeres, here is a cavity is formed which is termed as coelom. This cavity is termed as coelom, that is formed because of the splitting of hypomeres into somatic mesoderm and splenic mesoderm. This coelom is later on divided into embryonic coelom and extra embryonic coelom. Here is a close view of derm formation, in which you can see that here is the ectoderm that is formed by the remaining epiblast, while those epiblasts that migrate laterally through the primitive groove form mesoderm, in the result of which somatic mesoderm and splenic mesoderm is formed, and in between of this coelom is formed that is later on divided into two parts, two coelom, that is the embryonic coelom and extra embryonic coelom. This somatic layer later on attached with the ectoderm, in the result of which parietal layer is formed, while this splenic mesoderm attached with the endoderm, in the result of which visceral layer of body is formed. While here is the stalk is formed because of the, which is termed as yolk stalk, formed because of the hypoblast that connect the yolk with the embryo and supplies nutrients during the development. Here is the gastrocele that is termed as presumptive archantron or bodylet. These three terms later on form various various body organs includes hair, nails, skin, inner ear, lens are formed by the ectoderm. While mesoderm is divided into three regions, that is epimeres, mesomeres and hypomeres. Epimeres form axial skeleton, skeletal muscles, connective tissues of skin. While mesomere take part in the formation of reproductive system and urinary system. That includes oviduct, uterus, kidney, ovary and testis formation. While hypomere form connective tissue of body and legs by the parietal layer, while heart, blood vessels and blood cells are formed by the vessel layer of the mesoderm. While endoderm take part in the formation of intestine, liver, pancreas and lung. In this way, various organs is formed in the chick. I hope you got an idea about how various organs is formed and various terms are formed within the chick. This is take place by the process of gastulation. In the next lecture, we will study about the neuralization, how neurons are formed within the chick or nervous system developed within the chick. Thank you.